Hello, this is Tommy. Welcome to Chatomics. Today I'm going to show you how to do hierarchical clustering in R and understand what are the distance measures between data points and how you choose linkage methods when you are doing the clustering. So clustering is one of the unsupervised machine learning approaches, and it plays a very important role in any data analysis. So I hope after this video you can master how to do clustering in R. Okay, in hierarchical clustering, a dendrogram, so we first need to understand what is dendrogram. So a dendrogram is a diagram that shows the hierarchical relationship between the clusters that are formed like this. So it's a tree-like diagram that is commonly used to visualize the hierarchical clustering results. Okay, and the branch's length is signifying how similar the distance between different clusters. Okay, so we really need to understand, so before we do any clustering, we need to understand how the distance between data points are measured or defined. Okay, the different distance measurements, for example, Euclidean distance. So this is one of the most commonly used distance measure. So you have two data points here, the red one and the blue one. So x1, y1, and x2, y2. So if you calculate x1 minus x2 square plus y1 minus y2 square and take the square root, that's the Euclidean distance. For example, cosine distance, this one is this distance is measured by the cosine theta. The theta is the angle between those two data points. And for example, Jacob distance is defined by the intersection of two sets A and B divided by the union of those two sets A and B. Okay, there are many other distance measures. For example, Hamming distance, Manhattan distance, and Minkowski distance, etc. and etc. And in R, the distance usually can be calculated using the dist function. So if you question mark and dist to look at the help page, so you see, so dist, so first x will be the data matrix, so it will calculate the distances, okay, between I think between the rows of a data matrix. Okay, and now you can choose different methods. So, and the Euclidean distance actually is one of the default methods, and you can choose other ones here, for example, maximum, Manhattan, binary, and others. Okay, so after you define the distance measure, so we have to define how the clusters are merged, and that's what linkage does. Okay. Okay, there are many different linkage methods here. I listed 4 of them here. So, for example, single linkage. So linkage is a measure of dissimilarity between two sets of objects that are determining how two sets of objects are merged. Single linkage is minimal dissimilarity between points in two sets. And the complete linkage is the maximum dissimilarity between points in two sets of data points or two classes of the data points. The average linkage is a compromise between those two, so it's the average dissimilarity between points in two sets. And the what linkage is actually it minimizes the increase in the total within cluster variance when you're merging these two clusters together. So it is commonly used when the goal is to create a balanced and compact clusters. And in R, you can use the edge cluster function to do hierarchical clustering. So let's look at the help page. So the first argument will be the distance measure that produced by dist function we just covered. And then now you can choose different method of linkage. Okay, complete actually is the default here. Okay, but you can also choose single, average, and other. Okay. Okay, now we understand what is distance between different data points and what are the linkages. Let's use this NCI microarray data to practice how to do a hierarchical clustering in R. Okay, so let's load the library. So once you load this library, so this NCI 60 data set will be available in your environment, and it's a microarray data set for 60 different cancer cell lines. Okay, and NCI 60 is just a list and have two elements. One's the data, which is data matrix, and the other one is the label, which are the labels of each cell line. Okay, and if we first actually need to transpose it into a gene by sample data matrix. Okay, we can look at the first 5 rows and the 5 columns. Okay, and we need to add the column names, which are the labels, to the columns. Okay, so if you look at this dimension, so you have 6,830 genes, and columns are samples, 64 samples. But let's look at how many unique samples there. So unique, we have 14 actually different samples. Okay. Okay, so to do hierarchical clustering, the first step I usually do is to scale the data matrix, because some genes are highly expressed. You don't want them to dominate your clustering. So you want to scale the same gene across different samples. So scale really means, so x minus mu, the mean, divided by sigma, the standard deviation. Okay, so in R you can use the function called scale to do that, and you can specify whether you want to only center it x minus mu, or you want to do scale as well, x minus mu, and also divided by the standard deviation. And because scale function works on the columns, and we want to scale by rows, we need to first transpose this NCI data, this data matrix, scale it, and then transpose it back to the scaled matrix. Okay. Okay, let's first calculate the distance. Okay, use the dist function, and we already scaled the data matrix, and now because dist actually calculate by rows. So and now we want to calculate the distance between samples, which are columns. So we want to actually transpose it. So dist. So the matrix for the data distance will be dmat. It's a pairwise actually distance matrix. Okay, like this. Okay, so now we can do hierarchical clustering using hclust function, and let's apply this distance matrix and use complete linkage first. Okay, and complete linkage often gives comparable cluster sizes, less sensitive to outliers, works better with spherical distributions. Now we can use, so after we do the hclust, let's run that, and this is a hclust object. Okay, and you can actually access different elements in this object by using $sign, and it has different elements in that object. So we can actually plot the hclust object as a dendrogram, as we just introduced in the very beginning. So using the plot function, and okay, and this is really the hierarchical clustering results visualized by a dendrogram. Okay, so this is nice. We have the labels for each of the samples, but a little bit hard to see whether the same cancer cell lines are clustered together or not. So let's make the dendrogram look a little better by coloring the labels. Okay, so to do that, we will use this package called dend extend. Okay, and we'll use a polychrome package for the colors. We set seed to make sure it's reproducible, the colors. And then let's just sample 15 different colors. And you can look at those 15 colors. Oh, remember, we have actually 14 samples. We only need actually 14 clusters. But this color palette will give you a white color, so I will actually remove that white. So I will remove this color, so I will use the rest of the colors to denote distinct cancer types. Okay, so that's why I'm removing this first white. Okay, now we can actually convert this edge cluster. We need to actually first convert this edge cluster object to a dendrogram object. Okay, so we can just use s.dendrogram. Okay, and if you look at dend, it says it's a dendrogram object with 2 branches and 64 members. Okay, so now we can use the color labels function from the dend extend package, and then use the colors. Okay, the colors will be actually depending on the dend labels. Okay, we also need to extract the dend labels from the dendrogram. Okay, so if we do that, and it will tell you for each branch what are the labels. Okay, now we can plot it again. Now it looks much better because now the same actually cancer type is labeled with the same color. So you can see, okay, whether the same cancer types they're clustering together or not. For example, those are colon cancers, those are melanoma, those are renal cancers, those are non-small cell lung cancers. Okay. Because we're going to actually label those branches very often in the following code, so let's make it as a code. So as a function. So essentially, it's the same thing. Just wrap here as a function. Okay. Okay. We just tried complete linkage. Let's try a single linkage. Okay. It can actually handle diverse shapes, but it's very sensitive to outliers or noise. Okay? It often results in unbalanced clusters. Okay? We can use the same edge cluster function, the same distance matrix, and then specify method equals to single. Okay? And we can use our newly created plotting function, plot dend, and to see how it looks like. Okay. Okay. So this is the results of the clustering, but using the single linkage. Okay. So you see, most of the clusters are making sense as well, right? Makes sense as well. So similar, the same cluster, the same cancer types are close to each other. Okay, let's also try average linkage. Okay, so it's a compromise between single and complete linkage. Also less sensitive to outliers because it's average. Okay, let's do the same thing. And we see the dendrogram changed a little bit, but I think majority of the samples of the same cancer type are close to each other. Okay. Okay. Let's also try the word linkage, the same data matrix, but specifying method equals word, and plot it. Okay. Yeah. So in my experience, for gene expression data, people usually use average linkage or word linkage. And now, so once we actually have the dendrogram, we can actually cut it and to determine how many clusters we want to end up with. So this is kind of arbitrary, so and it's kind of subjective. So for example, if we specify the height, like here, 120 as a height, we cut it here and see how many clusters we're going to end up with. Okay, so we can further plot this. So this 120. And we end up with 8 different clusters. So we can do this by using the cut tree function and specifying the edge class object and also the height you want to cut here. And you see, after you cut the tree, it will tell you, okay, which cluster of each sample will belong to, right? Oh, actually, total 9 clusters, not 8. Now we can actually table it and look at different clusters and for what are the sample cancer types there. For example, for cluster 2, you only have like 7 actually renal cancer. And for example, for cluster 1, you have 2 breast cancer, 5 CNS, and there are other cancer types as well. It's kind of mixed. And for this one, cluster 5, you have 5 from leukemia, and that's it. So kind of pure cluster. Okay. So I just showed you use Euclidean distance, right? But now let's also use other distance, for example, Manhattan distance, the L1 distance. And then you can do all sorts of combinations, right, with different linkage methods. Okay, the same thing, and also give you a little bit different looking of the dendrogram. Okay, we can even try other distance metrics which are not available in the dist function. For example, we can use the correlation as a distance. So if you use correlation of the data matrix X, it will give you the distance between different columns, and 1 minus essentially is the similarity between different samples or different columns. Okay, let's do that. So what we can do is 1 minus correlation of this original data matrix. So if you do correlation and NCI data, give you a pairwise correlation between different samples. Okay, and you want to turn this correlation matrix as a distance matrix, as a dist object. You used as.dist. Okay, now this is your new actually distance matrix. Now we can use this one and specify different linkage methods as well. Okay. So one of the things you might want to know is that for hierarchical clustering for this dendrograms, so for each of the branch here, you can flip it left to right, and it doesn't actually change the meaning of clustering, right? So for example, those two samples at this branch, you can switch this to here and here to here, right? So we can actually sort the dendrogram, okay? And just to make it a little bit more visual, okay? So we are going to use the dend sort function. So it will sort the dendrogram based on the average distance of subtrees at every merge point, okay? So the tighter the cluster, in other words, the cluster with some smaller average distance is placed on the left side of the branch, and when a leaf merge with a cluster, then that leaf is placed on the right side. Okay, let's do that. Use the load library, and we first plot the dendrogram. So this without sorting, but we can actually just pipe it into dendrosort and to sort the dendrogram. Let's see how it looks like. Now you see now, because the dendrogram now is sorted, so it looks a little bit better. Okay. Yeah, I think that's it for today. So I hope you understand what is hierarchical clustering, how to do it in R, and what are the distance measures and what are the linkage methods. And there's really no correct or quote unquote correct ways to do clustering. So different distance measures you can use and different linkage methods you can use, and really depends on your data. And really a clustering is an art, so you really want to interpret it in the context of biology. Okay, that's it for today. I hope you enjoy it, and make sure you subscribe to this channel, and I will see you next time. Happy learning.